Now, you should never Google your heroes, because you find things that you really don't want to know about sometimes. However, in this case, Julian Alexander Hiscox is a qualified instructor in Wing Chun Kung Fu. The practice of this ancient art will give you energy, confidence, and make you new friends, which is evident today. He also leads the Hiscox Lab at the University of Liverpool and is Deputy Dean in our Institute of Infection, Veterinary and Ecological Sciences. So I will give the floor to our host. Thank you.
Thanks, Callum. As this is actually live, I was stopped being Deputy Dean a couple of years ago, and it's Nicola Williams. Yeah, I'm now Deputy Claire Ayres now. Always Deputy Dog. But the Kung Fu thing's correct. Thank you. That one.
I think not. Perfect.
So I'm following on from a series of lovely talks looking at antivirals in feline, in cats. So I thought I'd talk about some of the work we've been doing with many other people and led by many other people on evaluation of antiviral drugs for SARS-CoV-2, but also looking at how we can better track the future evolution of SARS-CoV-2 and potentially other coronaviruses and other viruses in general. So one of the things we've been obsessed with in my lab for probably about 14 years now is this idea of viral quasi-species or viral population genetics, depending on who you talk to and whether you come at it as a virologist or a mathematician. I always get corrected. I'm never too sure which one I'm meant to use. But when you sequence a virus from a clinical sample, what you generally end up with is something called a dominant or consensus sequence, which represents the majority viral population in that sample. And that is generally what gets uploaded to global databases such as GISAID and so on. But underneath that is an awful lot of what we would consider genetic noise, but it's that genetic noise that evolution and selection acts upon. So we've been looking at this in collaboration with James and Al in the audience and many others over the years, both for coronaviruses and filoviruses, including Ebola. And the two things I'm going to talk about first were led by a couple of PhD students in the laboratory, Aya Donovan Banfield on the molnupiravir, Hannah Goldswain on looking at evolution in quotes, normal people and test prints for postdoc in the lab for some laboratory work, and finish up with some, I guess, esoteric work done by Xiaofeng Dong in my lab on how can we predict the future evolutionary space of viruses. So one thing to remember with SARS-CoV-2 and other coronaviruses is that in an acute infection, so that's an infection that doesn't last for very long, the primary dominant driver of genetic change are single nucleotide polymorphisms, either done by the viral polymerase or host cell factors. Of course, there's an emerging theory that as you get a persistent infection with coronaviruses, then this gives the opportunity for indels and recombination to flourish to enact genetic change. And early in the outbreak and throughout the outbreak, we've worked very closely with Callum and Isaric. So to give you an example of that, I guess the first major change that occurred in SARS-CoV-2 that would have been called a VOC had we have had the phrase VOC at the time was the D614G substitution in spike and P323L in the viral RNA-dependent RNA polymerase. These emerged early in 2020, right at the start of the pandemic. So when we took Isaric collected samples from the UK, you could see a rapid emergence of these two variants, or of this variant containing these two amino acid substitutions, from a minor variant level in the population into dominant consensus sequence. And you can actually look at the speed of some of these changes in a non-human primate model that we did, led by my long-term friend and collaborator, Miles Carroll, where if you start with a viral population in a non-human primate that is predominantly P323 or predominantly D614G, within a matter of days in those non-human primates, you see a complete change to the 323L version of NSP12, telling you that that substitution is under immense selection pressure. And also using ISRIC data as well, we can see it in individual patients. And in actual fact, when we did reverse genetic studies with our friends at Bristol, Andrew Davis and David Matthews and their laboratory teams, one of my PhD students, Mohammed Al-Rali, and postdoc Stuart Armstrong did a lot of pull downs with the two different variants of NSP12. What we actually think is going on is that this is a host adapting mutation in the virus. But the point of this is that these mutations or substitutions started off in the minor variant population in humans and then were rapidly selected for within the host, and then were obviously transmitted from host to host. So during the outbreak, we were and still are involved in the Agile clinical trial platform that is led by many people, but two of them are Professor Sei Koo at the University of Liverpool and Dr. Tom Fletcher at the Liverpool School of Tropical Medicine. The Agile platform is to develop and roll out rapidly antivirals to emerging zoonotic or emerging pandemic threats. Because obviously in the time of pandemics, you can't take 10 years to do your drug trials, because at the end of that, everybody's dead and we're all turned into zombies. So you've got to bring your antivirals out very, very quickly. And so they came up with this new way of doing a drug trial, and the role in our lab and with in collaboration with Al Darby was to sequence samples from this clinical trial, identify a potential mechanism of action, in this case of molnupiravir, which we already had some ideas of, and then as a scientific curiosity, not part of the trial, to see whether we could identify mutations or substitutions that were associated with resistance. So molnupiravir is effectively like a mutagen. We've done a lot of work historically on respiratory syncytial virus and ribavirin and other antiviral drugs for Ebola, so could put this through several different pipelines. And one of the things you would predict with molnupiravir is that in SARS-CoV-2 genomes exposed to molnupiravir, you will see an increase in the TS-TV ratio. And indeed, when you look at your patient groups that were either placebo-controlled or treated with molnupiravir, then you see a difference in the SARS-CoV-2 in patients treated with molnupiravir, in that you see an increase in this TS-TV ratio that confirms that the drug is working and it is mutating the virus, and hopefully mutating it towards error catastrophe. And one of the things that we did in the lab is follow that up with in vitro studies, where we were passaging the virus in molnupiravir sequentially to see whether we could see any resistance. And effectively, we couldn't see resistance to molnupiravir. What you do see are genomes that survive molnupiravir treatment. And Aya, with groups at Cambridge at the Crick Institute, also examined molnupiravir remnant genomes, as I call them, in the human population that you see post-molnupiravir treatment. So the virus still survives treatment with that antiviral. I think what this tells us, and James will follow up with this after the break, is that if you give an antiviral drug, you have to give it at the beginning of infection or even prophylactically, and that's a very easy thing for me to say. It's what we follow with influenza virus, but if you give molnupiravir too late, then you've still got replication of the virus going on and damage associated with the virus. So this kind of got us to thinking about, are there evolutionary pressure points in coronaviruses? Can we think about predicting the future evolution of the virus? I was brought up on TGV, transmissible gastroenteritis virus, and like Zoltan in the audience, infectious bronchitis virus. You see a massive diverse array of genotypes and serotypes. This is driven by single nucleotide polymorphisms and recombination. There's no universal vaccine for IBV. The vaccine needs continuously updating. Your diagnostics needs changing, blah, 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 blah. And Callum and I said much of this advice at the start of the pandemic to mainly deaf ears, that we're going to have to change the vaccine for SARS-CoV-2 because coronaviruses, as you all know, change. But being able to predict those changes might be able to get you ahead of a particular curve in a pandemic. It will allow you to do longer-lived, and I'm mainly funded by the Food and Drug Administration in the States, longer-lived medical countermeasures, so vaccines, antivirals, and diagnostics. And we got to thinking around this when Xiaofeng in the lab went back and examined publicly available sequencing data for workers in the Huanan seafood market, the epicenter of the COVID-19 pandemic, where when we actually looked at their sequencing data, we could identify hallmarks of future VOX already present in the minor variant genomes in those patients. And of course, for those of us of a certain age, we will be familiar with the phrase hypervariable region in a virus glycoprotein. And that is where you compare consensus sequences. RSV is a classic for this, where it will have a number of sites that vary. But what we're looking at this is this variability in minor variant genomes. So in the minor variant genomes in people, you've already got variability in amino acid sites in the viral proteome or genome that are hallmark of future evolutionary pressure and selection. So one of the things we did in the UK, and Al spoke about this yesterday and was the deputy director of it, was COG-UK, where we sequenced an awful lot of samples from people with COVID-19. And what you're looking at here is a diagrammatic representation of variation in the spike glycoprotein at an amino acid level, for example, on the left-hand side between March and April 2020. No variation whatsoever, or very little. Then, of course, if you compare March 2020, say, to May 2021, then that's where we're starting to see VOCs associated with alpha and delta becoming dominant in the minor variant genome population. Now, I remind you that we didn't see delta till a little bit later on in the pandemic. But what we did is that we derived consensus genome, minor variant genomes, for 100 patients per day for nearly 3 years computationally using the COG-UK data. One of the key things in this is this thing called a W value. And each time I explain this, I think I get a little better at explaining it, but the concept is this. If you imagine that you sequence a virus from someone, imagine there is no genetic variation in that virus, and there are no sequencing errors, then you will get 100% frequency of each amino acid along the genome. Now, of course, in real life, we get sequencing errors. And if you get sequencing errors, the variation at those amino acid sites will be slightly different because of the inherent bias in sequencing. So rather than being a W value of 1, it will be, say, 0.99957 or something like that. And then, of course, you get errors due to the error-prone replication of coronaviruses, and you get errors due to the action of host cell proteins as well, all acting on the genome. And again, that will alter the W value. Now, if you get selection pressure acting on those amino acids and those sites in the genome, then your W value will decrease from 1 and tend towards 0. And then you can think about that occurring then in a population. So that W value, if it's undergoing selection pressure, will shift from the left-hand side here over towards the right-hand side if a site is undergoing selection pressure. And so if you then compare that with your variation frequency, you end up with a molecular map of variation in a particular protein in a particular virus. In this case, this is SARS-CoV-2 spike glycoprotein. Each dot there on the right-hand side represents an individual amino acid position in spike. And what you see at the top there is a clustering of amino acids that are not undergoing selection pressure. And then coming down from right to left are amino acids that are, for want of a better phrase, being offered up for selection. And then on the right-hand side are amino acids that have become selected in your dominant viral population. In this case, at this particular time, it was amino acids in spike associated with our homegrown alpha/Kent variant at the time. But in those things, in blue, you can already see that you can see amino acids associated with the Omicron VOC, even though Omicron had not been identified at that time and emerged from southern Africa. So that genetic promiscuity of the virus, you are getting specific changes at specific amino acids and specific parts in the genome that can tolerate change. And so this raw material of evolution is being percolated already, even before those amino acids are being selected. So we put together a little movie of this. And this is now in May 2020, but moving onwards into June 2020. And you're looking at spike, the spike glycoprotein. Each dot is an amino acid in spike. And as things are offered up for selection, as they become selected for, they move from this pool in the middle over towards the right-hand side. So in red, you've got your amino acids associated with the alpha variant becoming dominant, but you can see that things to do with the Delta variant, even though Delta was still in India at the time, are moving upwards for selection. And then, of course, as we move into the wild times of Omicron, we can see those Omicron amino acids becoming dominant. But the point is that this genetic variability, even though Omicron existed, it came from southern Africa, so we think, that genetic variability is being offered up for selection in the UK population. And I'm sure it would be the same in any other sequencing that we did of SARS-CoV-2 globally. So then what we did is we used machine learning to take these variation frequencies and these W values and group them into clusters. And so we did that, say, for the first 3 months, 6 months, 12 months, 36 months of COG UK data. And again, this is just some diagrams of spike. We've looked at other SARS-CoV-2 proteins as well. But for example, if we look at our model called PAM12, this is the first 12 months of COG UK data for spike. It clusters into 3 distinct regions in grey, orange, and blue. And in blue are the amino acid positions in spike that we think are most likely to be most variable. Ignore the red dots for a minute and imagine you go away in a time machine for 2 years, or you go into suspended animation or some type of time dilation, and you come back 2 years later and you say, okay, where have the changes in spike occurred based on our first 12 months of modelling data? In actual fact, most of the changes occur in the blue region. We can then take nearly 3 years of COG UK data and then say, okay, where are our changes occurring in that? So about 75% of the future changes that occur where we think they're going to occur. And of course, we can then start mapping this onto the structure of the spike protein, and this is done in collaboration with Jules Gallo and Alan Bailey at the Pearbright Institute. And our PAM34 model, based on the first nearly 3 years of COG UK data, correctly identified 13 out of the 15 changes seen in the most recent variant of SARS-CoV-2, JN1. So the models are very good at taking this genetic variability in sequencing data and then saying, where do these changes occur? Where are they most likely to occur? So how do we want to think about this in terms of the CEPI 100-day pandemic mission? Can we use this information to make rational vaccines that don't need to be changed, or diagnostics that don't need to be changed, or sequencing primers that don't need to be changed? So we're currently looking at whether, for example, 1,000 genomes per day for the first 3 months of the pandemic will give us a suitable model, or do we need 10,000 genomes for 3 days? Those types of parameters. And one of the other things that we're doing is to see whether we can improve our machine learning approaches, but also together using reverse genetics approaches and pseudoviruses, looking at why some amino acids that are offered up for selection in pairs or whatever don't become fixed. Why didn't they become selected? What is it about them that allows them to remain stable in the genome? Okay, I'll end by acknowledging all of the lab, and it was very much all a group effort during pandemic times and still is in the laboratory. Our funders, and particularly our relationship with COG UK and ISARIC as well, that allowed us to get these really precious samples and data and to do some research on them. So thank you for your attention, and I'll take any questions.
Very nice, Julian. Before you showed the structure of the RBD, I was thinking, okay, can you connect the genetic data to functional, at least some hints what this means in functional terms? So how far are we away from predicting if mutations are are reflected in a structure of a protein, and particularly outside the spike, you know?
Yeah, so it's a nice question, Volker. So we've got two funding pathways in the UK, one called G2P2, which James and I are part of, and the other one called PROVAC. And one of the things that they're doing is trying to look at this idea of future evolution. And a lot of people are using a deep scanning mutagenesis approach. So I think the idea is that we would take this together with the deep scanning mutagenesis approach, and then use things like pseudoviruses to rapidly screen for neutralization or infectivity. And so you could risk assess the changes.
I think...
Go on, I can hear you, Volker.
I think risk assessment is one thing, but now we have this huge database, you know, of sequencing data from now 4 years.
Yes.
It's maybe good to look back and find out why have particular mutations been selected, and what do they mean in terms of function, you know?
Yeah, so I think that's part of G2P2 is doing that, but I guess our role in that is to try and guide that analysis using machine learning to pick more likely candidates, and we do that from the sequencing data. We tend to use COG UK data because it is very defined data set. We know the sequencing strategies that we used, and we can correct the errors for those. Lucy.
Thank you. That was really amazing, really fascinating. And this is probably an extremely naive question, but I just wondered, how much is this machine learning model trained on the fact that there was a lockdown and we were all behaving in a completely different way to how we might be expected to behave in future pandemics because nobody will want to lock down? Do you think that's got any influence on how certain variants rose to the top?
I think that's a good question, and I think actually when you talk to people, they actually behaved as they were going to behave during lockdown, and a lot of people snuck visits to other people. Anyway, certainly in my friendship group, I mean, I think I was Mr. Squeaky Clean, but I know there was a lot more genetic mixing than what we thought went on. But the thing about, you're right, but the thing about the COG UK data is that you've got all the sequencing metadata associated with it. So this work was done using NovaSeq data, using the Arctic pipeline. Now the thing is that would it work for like the MinIONs that I'm a bit obsessed with? And you're right, I think one of the two things we want to do is that we did a lot of sequencing as part of the European Mobile Lab for the Ebola outbreak. We've got another 2,000 samples we can sequence. Can we do the same thing for EBOV? We work very closely with my friend and colleague Waleed Al-Jabbar and others in Saudi Arabia for MERS. Sample set's less, but given that with the Huanan market, you had that picture. Can we use, less samples? But you're right. Al, you can't ask me anything nasty.
I'll have to rethink now. So I was just thinking that one of the things that we haven't done is we haven't mapped those SNP proteins to see if they're linked to other genome SNPs,
Yes.
have we?
Yeah.
And this is one of those things that I think others in the room might be interested to look at now, is that you'd expect there to be epigenetic effects across the genome, so that although those mutations were there, maybe there wasn't the right genetic background for them to be selected in the way that we saw later.
So that's a great point, and I'll mention his name in case he's listening in. Greg Towers at UCL made that same point to me, and Greg did some lovely work with Lucy Thorne and lots of other people where they were showing that when you got a change, say, in the, keep calling it ribosome binding site, receptor binding domain in spike, it might alter the infectivity of the virus, but that would be made up by a compensatory mutation in one of the accessory proteins. And then, of course, Omicron is relearning that. So you're absolutely right. I think that's the next thing to get Xiaofeng to do, is to look for linkage and association.
Yeah. But, I mean, I think for people starting off at this point, if they've got the data sets to try and do that, it highlights the value of whole genome data, I think. That's what I'm also trying to be evangelical about.
Yeah.
So that was a nice question, yeah?
All right. We'll go to Peter now.
I absolutely loved the cartoon with the movement of the amino acids and the selection. My question, because molnupiravir targets the RNA-dependent RNA polymerase, and presumably the mutations we see are in the catalytic side, are they?
No.
Do they have an effect on the replication efficiency of the virus?
Yes, no and yes. So molnupiravir is just incorporated by the RNA-dependent RNA polymerase. I think one of the previous speakers mentioned Paxlovid, and I think James will talk about Paxlovid as well. That actually targets an activity of the replicase complex. All the RNA-dependent RNA polymerase, it just thinks molnupiravir is a nucleotide. It can't see it as anything else, but it gets incorporated in, and then as the virus replicates, will get changed to something else. So you end up getting loads and loads of errors along the genome. What we don't see are any, and I can't remember the precise phrase Say taught us to use, is that we don't see amino acid substitutions that are enriched in the presence of molnupiravir that would be indicative of a resistant phenotype emerging.
Interesting, because, you know, thinking of the hepatitis B and lamivudine, for example, when those mutations occur, the mutants are less fit.
Yeah.
The minute you withdraw the drug, they disappear and the wild type comes up again. Obviously, this is not happening here.
What you do see, and this is the work of others, we see it in cell culture, but they actually looked at this in the human population, are what I would call molnupiravir remnant genomes being present and spread in the human population.
Well, ladies and gentlemen, thank you for a very, very illuminating session.